Hello everyone, and welcome to today's webinar, SPNTypeID, a framework for bioinformatics workflow automation. I am your host, Dr. Abigail Shockey. I am the bioinformatics training lead at the Wisconsin State Lab of Hygiene's Communicable Disease Division. And today I'm going to be talking about the way in which we fully automated our Streptococcus pneumoniae serotyping pipeline using various infrastructure components to give you guys an idea and a framework of how you can automate your own bioinformatics pipelines. So who is this presentation for? This presentation is primarily designed for bioinformaticians who are interested in automating their workflows using cloud computing, particularly for the purposes of validation, because our Streptococcus pneumoniae pipeline is validated. To get the most out of this presentation, you should have the basic understanding of a coding language such as Python, a basic understanding of bioinformatic workflow development, and an understanding of cloud computing. As for what you'll learn, today we're going to discuss the key components of our cloud infrastructure that allowed us to automate our Streptococcus pneumoniae serotyping pipeline, which is called SPNTypeID. These aspects will be discussed broadly to allow for translation across other cloud computing platforms. I'm going to be talking a lot about AWS, Amazon Web Services, and the pieces of AWS that we use for our cloud computing infrastructure, but I'm going to talk about it in a way that hopefully if you have a cloud computing platform other than AWS, you might have a tool equivalent in it that would allow you to do something similar. Now, here's an outline. First, I'm going to give you an overview of the workflow. I'm going to be talking about how sequence data is transferred, the actual launching of the workflow, post-workflow management steps, and of course, conclusions. So, the workflow itself. The purpose of the SPNTypeID workflow is for the genome assembly and serotyping of Streptococcus pneumoniae. It's written in the workflow management language Nextflow. According to the NF Core standard, its input is paired raw Illumina reads, and the output is a report containing QC metrics and the predicted serotype. As for the availability of this code, all code is open source and available on GitHub. You can see in the bottom left-hand corner right here that there is a URL to that GitHub repository if you were interested in the Nextflow architecture that makes up the workflow. This is separate from the cloud infrastructure that we've used to automate the workflow, but if you're curious, you can take a look at it there. So this is a diagram of SPNTypeID, specifically version 1.6.1. I'm not going to go through every single step in this diagram, but I do want to highlight different parts of it. Here we have the input I mentioned previously, which are raw Illumina reads that are paired, colored in gray. Here we have these various analysis steps, such as serotyping, its main purpose, genome assembly, read trimming and cleaning, calculating coverage and other quality statistics, and the classification of genus and species, things such as that. And then we have these summary steps, which take the results of the different analysis steps and summarize them so that they can go into the final report. Now, how is SPNTypeID automated? As I previously mentioned, we use a number of Amazon Web Services to allow for SPNTypeID's automation. These services include Lambda, which performs tasks, Batch, which provisions computing resources, EC2, which is the actual cloud computing component, S3 for storage, DataSync for transfer, and DynamoDB, which is their database service. So I'm going to go into these services just a little bit more in depth. Again, I'm keeping it pretty broad. AWS Lambda is a compute service that runs code and is used to perform small tasks such as moving data between storage buckets and launching workflows. AWS Batch is the computing service that supports workloads of any scale. It automatically provisions computing resources and optimizes the workflow distribution based on the quantity and scale of the workloads associated with the workflow. AWS EC2, which stands for Elastic Compute Cloud, is an on-demand scalable computing capacity in AWS Cloud. S3, which stands for Simple Storage Service, is an object storage service that offers data storage that is scalable with high availability, security, and performance. AWS Data Sync is a service that allows the secure transfer of data from on-premises network storage to locations in S3. And DynamoDB is a serverless database service in which data is organized into tables. Now, you'll notice that there are icons associated with these. I do have diagrams of our cloud computing infrastructure that I'm going to show you, and these will feature these icons. So there's some additional parts of this in how it's automated. All of the cloud infrastructure is managed through code rather than manual processes, and this is through the infrastructure as code tool Terraform, and the pipeline is managed, launched, and monitored using the Sakara platform, also known as Nextflow Tower. There are actually only 3 folders our sequencing team members interact with. The submit folder, which is where FASTQs are moved by our sequencing team, the sample sheets folder, which is where sample sheets, which I'll talk about this in a minute, that are automatically generated are synced, and a results folder where results files are synced after the workflow has finished. So this is just a screenshot here of the directories on a Windows machine that our sequencing team members would see. So there's 2 diagrams I'm going to show you about this infrastructure. The first is the Nextflow analysis architecture. I'm not going to go through every single part of this now. I'm going to go through it in stages. I just want to give you this as a highlight, but this is the actual infrastructure through which the Nextflow workflow is launched. And then we have the workflow automation architecture diagram, which again, I'm going to go through. The things you need to know for now is that anything that is on premises at WSLH is in a gray box. We have this little key here that shows when something is done through API messaging, and we have these solid lines here to indicate data transfer between services. And then anything that happens in AWS Cloud is shown here in this darker gray box. And so first things first, I'm going to talk to you about how sequence data transfer is automated. And the key component of that is data synchronization. This uses data sync, and a data sync task will synchronize completed sequencing data from an on-premises folder to an S3 bucket. So like I said, our sequencing team members will take reads and put them into a folder, and then a data sync task, which is performed every hour on the hour, will sync sequence data up into the corresponding S3 bucket in AWS Cloud. And the equivalent for this, if you weren't using AWS, is simply a tool that would transfer sequence data from an on-premises location to cloud storage. And this is this part of the diagram here in this red box, where we have genomic data present on a drive at WSLH, and then a data sync task here transfers data to what we call our genomic data inbox, which is in AWS Cloud. So now we can move on to the workflow launch itself. The first step in workflow launching is the sample sheet generation. Nextflow workflows take in sample sheets as input, and we use a Lambda function that generates a sample sheet containing sample IDs and the full path to the corresponding FASTQ files in S3 storage. So once reads have been moved via that data sync task, a Lambda function will create a sample sheet with the sample IDs and the FASTQ files to the S3 storage bucket they were moved to. Once that is done, the Lambda function will launch the SPNTypeID workflow. All parameters associated with workflow execution are collected from Nextflow Tower via an API. And these parameters include things like workflow parameters, input, output, and working directories, which will all be S3 buckets since the workflow is performed on the cloud. The compute environment ID, so whether or not it is our development or production environment, because we do have 2 separate environments. One where if we are performing an analysis, whether it be automated or manual, if it was one of our other pipelines, we perform those in our production environment. But if we are testing changes to our pipelines, we do those in the development environment. And of course, AWS access keys, which are credentials that AWS uses that give us access to our computing environments. Now, workflow runs are submitted to AWS Batch, which scales the deployment of EC2 instances to meet the computing needs of each run. And the requested EC2 instances are created within a virtual private cloud environment. and data is transferred into and out of the EC2 instances and S3 buckets. And again, I'm going to show you on the diagram what that looks like. So when it comes to tracking sequence data analysis, a Lambda function will update a DynamoDB table to track which samples have been analyzed so that we don't accidentally repeat analyses. This is where this part of the workflow automation occurs on the automation architecture. We have our genomic data inbox here, which is the S3 bucket the data sync task moved the sequence data files to. We have our Lambda function here, which is the workflow launcher, as well as DynamoDB, which will update if an analysis has been run. And then we have actually the Sakara platform come into play, which is communicated with through an API. And then we have the architecture here for running the workflows, and the results of the analyses are moved to what we call the genomic data analysis bucket, which will eventually go on to the next step in the workflow automation, which I'll talk about next. Now, as for the Nextflow analysis architecture, it'll give you a little bit more clarity here, where we again have that AWS data sync task where we have the genomic data inbox and the workflow automation. But this is where EC2 and batch come into play, where batch will provision the necessary computing requirements, and EC2 are the instances where the analysis is run. So the necessary information about computing resources is given to batch via an API from Nextflow Tower, and the actual runs are performed in EC2. And again, anything that comes out of an analysis is dropped into the genomic data analysis bucket, which is an S3 bucket. Now, as for workflow launch equivalence, you would need a method for scanning where your sequence data is stored, as well as a method for updating a database that tracks if the sequence data has been analyzed so you don't have any repeat analyses. You would need a method for determining which workflow to analyze the sequencing run with. In our case, we have a separate Lambda function that only triggers strep pneumo analysis, but you could have another way of tracking what workflow to use if you had multiple workflows. You could use something with like test IDs that you would store in a laboratory information management system. You need a tool for generating your sample sheet if you're using Nextflow. You need a tool for launching the workflow once the sample sheet has been made, and you need the computing infrastructure to run the workflow on once it's been launched. Now, as for post-workflow management, you need to be able to monitor workflow completion. And so a Lambda function will monitor workflows by parsing the Nextflow log file for particular lines. So when a Nextflow workflow finishes, it will update its log file with this statement, workflow completed greater than workflow stats. And this indicates that the workflow is finished and the output files were transferred to their designated output directory. We also have a data sync task that monitors results folders, and when data is present, it will sync them to the on-premises results folder. Now, as for organizing output files, we also have a Lambda function that organizes specific workflow outputs into pre-designated S3 bucket locations. And as I said, we have an AWS data sync task that transfers output files from S3 storage to our on-premises folder for our sequencing team to retrieve. So this is this section of the diagram where, again, we have results data that has been stored in our genomic data analysis bucket. And then we have our post-analysis automation, and that Lambda function will put any files we want to organize for long-term storage into our genomic data results bucket. And then we also have our data sync task, which will take results from the genomic data analysis bucket and the genomic data results bucket and put them into on-premises storage. So for post-workflow management equivalence, you would need a method for determining when a workflow has successfully completed, a tool for monitoring workflow completion according to that designated method. You would need a tool for organizing your output files, and a tool to transfer results from cloud storage to an on-premises storage. So in conclusion, as you can probably gather from this presentation, workflow automation is possible with the right combination of tools, but the process is quite complex and has many moving parts. And cloud computing can simplify automation as different services from the same provider are designed to interact with one another. It would be very difficult to do this automation if we didn't have as many AWS services as we have access to that can all talk to one another and facilitate the automation of this workflow. I would like to give acknowledgments to the CDD bioinformatics team and sequencing team, specifically those members of the team that are highlighted here who have made a lot of contributions to the workflow automation, and then of course the members of our sequencing team who actually are in charge of the sequencing of our samples that are analyzed by this workflow. And of course I have to give a funding statement. This presentation was made possible through funding provided under the Epidemiology and Laboratory Capacity for Prevention and Control of Emerging Infectious Disease, otherwise known as ELC, Cooperative Agreement, Project E, Advanced Molecular Detection, to the Wisconsin State Laboratory of Hygiene. The conclusions, findings, and opinions expressed by authors do not necessarily reflect the official position of the U.S. Department of Health and Human Services, the Public Health Service, or the Centers for Disease Control and Prevention.